Uh, my my name is Phan Luu. I'm a scientist at the Brain Electrophysiology Laboratory Company, and today's talk really is an extension of work that we've published last year. We being uh, Don, Carl, and I. In this talk, we go a little bit more into the prosomeric model and how it can account for the active affordance basis, really, of active inference, the motivation that drives exploration and exploitation. This is the talk outline, a brief introduction into motor control and visual perception, and then we'll step through what the prosomeric model is and what that implies for the neurohierarchy. And then we look at cybernetic control, feed forward and feedback systems that underlie or reflects different systems of active affordance. And then the evidence for them. If we have time, then we can go into the catecholamines a little bit. This is a somewhat of a lot to cover, so I'll try to cover it from a high level to give a overall understanding of our approach to this topic. Okay. So for a long time, Don and I have been interested in brain evolution and motor control. That is the cybernetics of motor control. And this really started in the 80s with the the evidence of dual trends, particularly in visual processing. That's on the left-hand side here. This is based on the work of Ungerleider and Mishkin, wherein they show that visual processing, if you trace it from the occipital lobe here, there seems to be two paths, a dorsal medial path and a ventral lateral path is involved in visual processing. The ventrolateral, I think this is quite well known now by scientists in the field, of visual object processing and spatial processing, dorsal being spatial, ventral being visual. And actually, an updated version of that it has motor vision and motor implications all sort of tied together with these two streams. About the same time, Gary Goldberg, examining the evidence of human lesion, animal data, and so on, noticed that there seems to be something similar in the frontal lobes. That there are two paths of motor control, really, originating, and this is important, originating in the limbic cortices. And really, he based his theory of motor control on the idea of limbic control, tracing it back to evolutionary basis of the cortex based on Sanidi's work, wherein the cortex reflects the development of the cortex, evolution of the cortex, reflects a development from the hippocampus or the archicortex and the paleocortex or the olfactory cortex. Olfactory being ventral and lateral, and archicortex being medial and dorsal. And that motor control takes on two forms. One is a feedback form, where action is guided by the sensory input, particularly visual, as an example, like grasping an object. And the dorsal, mediodorsal system is involved in a primitive form of planning where action is internally generated as opposed to externally guided. There's an impulse, there's an urge, and action is launched into the environment, into the world, and it complements the Unger, Leiter, and Mishkin model of visual perception. So again, I want to emphasize the important limbic basis that Gary Goldberg established or proposed. So, and this model, this model of cortical evolution, was proposed in the late 60s, early 70s, and it sort of fell out of favor for a few decades. And then as scientific methods evolved, um, uh, the prosomeric model was developed to explain the organization and development of the brain. And so here, let me describe what the prosomeric model is. So this is the decomposition of the neurotube of amphioxus. And this is a cross section on this side of one of these segments. What you can see here are zones, basically. This is the top, this is the bottom, this is the floor plate, and this is the roof plate. These zones secrete morphogens. Those are proteins that guide development of cells, of neurons. And on the lateral side, we have the basal plate and the alar plate. This is a flattened diagram of the neurotube. This is the top. This is unfolded, so this would be the roof plate. This is the plate. And these are the alar and basal, so it's just flattened out. So how these segments are organized is based on the expression of genes and their proteins, and cell proliferation, and so these so in amphioxus there are three parts: the hypothalamus, the thalamus and midbrain fused as one neuromeric unit, and then the rest of the hindbrain and spinal cord. Uh, and so we can already recognize features of the vertebrate brain, particularly all of these components. But what is interesting is there is obviously no telencephalon, no cortex, no subpallium, no striatum, and no thalamus proper as well. So it's all fused. So the prosomeric model is not only a organizational model, it's a developmental model, as well as it has some functional implications. And this requires a little bit of a history of different models of the brain. Herrick in the early 1900s had his model today referred to as the columnar model, because he had these zones that started in the hindbrain here, and they were defined basically from data of nerve endings and nuclei associated with sensory and motor control in the spinal cord. And there are four zones, longitudinal zones, that go from back to front. There's the somatosensory at the very ventral or dorsal aspect, somatomotor at the very ventral, and then visceral motor and visceral sensory in between. Again, this is a description as well as a functional implication of this organization. And it extends, and he extended it all the way into the front, into the prosencephalon. And then in the mid 1900s, other scientists had an alternative model of organization and development. And this was based on evidence of during development, there were grooves and bulges in the neurotube. And the thinking was that these grooves and bulges, because they have cells that start there and proliferate there and then migrate out, that based on these features of bulges and grooves, that they make up units, and these lines show the units. In some ways, it's consistent with Herrick's columnar model. There are four longitudinal zones, at least up until this point. There is a line that goes actually from back to front. That is not unlike one of these going back to front, but and then there are additional units that are cutting in this the coronal plane. So that's called the synthetic model. And then in about the 90s, when we had the molecular techniques, and this is mainly based on the work of Puelis and his colleagues, where they pretty much confirmed this general organization of the synthetic model, but had more components, more units, and extending the refinement all the way into the prosencephalon. And these borders are marked by the expression of different genes and proteins. And so the particular insight here is that if you look at the spinal cord as an example here, so again, this is the floor plate, roof plate, alar domain, basal domain, and you look at the expression of certain proteins and genes in the spinal cord. There are two or a class of genes and their proteins that direct the development of cells, neurons. In the ventral domain, the floor plate, it expresses SHH and the proteins in a gradient that goes from ventral to dorsal, ending at this border. The roof plate, on the other hand, expresses WNT, BMP in this gradient, and they oppose each other. But even in the spinal cord, we can start to see things that will be repeated in the forebrain, and so SHH is is important to the development of GABAergic neurons. Whereas these are important to the development of glutamatergic excitatory neurons, and that we'll see repeated up to the cortex. So here is a picture then of the neural organization in fetal development. So this is this bulge here is the telencephalon. That's the cortex and the basal ganglia and other structures like the amygdala. And you can see the floor plate here and the roof plate. And the floor plate, again, expressing SHH. And then there's this other precordial plate that extends basically SHH signaling of the floor plate into the front of the brain to really influence the development of GABAergic neurons in the forebrain in this direction. Another sort of important fact based on the prosomeric model is the origins of the cerebral cortex, or the telencephalon. It comes out of the hypothalamus. So this is one component of the hypothalamus, the terminal hypothalamus, and then this is the second component. It is this neuromere, this section that gives rise to the telencephalon. So the telencephalon actually has a hypothalamic basis. And again, these arrows just show the direction of morphogen, the gradient direction, and they oppose each other. Okay. So let me just pause right there to see if there are any questions. Okay.
We usually do this after you finish the presentation in the Q&A.
Okay. So now, if we take then this folded and model or schematic of the developing brain, and we flattened it out according to see the relationship between different parts of the developing brain. It provides us an insight into how the brain is organized that is much clearer than looking at something that is folded and contorted during development. Here, so this is the back. On this side, this is also the back. So the front of the brain is here. And this is the alar plate, basal plate, and this is the this border here marks the border of the telencephalon. So you can see again here the hypothalamus giving rise to the striatum and the basal ganglia and the cerebral cortex. So this is a front to back. And notice that the basal ganglia sits in the front of the cortex, not below the cortex, like you would find in a folded model or in actuality. And you can see here the gradient of SHH being dense here and diffusing across here. And then it is opposed by the morphogens, the proteins in the cortex to limit the development of GABAergic neurons to this domain and glutamatergic neurons to these domains. Okay. And so looking at just just this, the cortex itself. So here, I'm going to zoom in on it. So again, you see within this, within the cortex, there are additional patterning centers. So we had the roof plate and the floor plate. Within the core of the neurotube, within the cortex, we now have what are called tertiary patterning centers. The hem, which sits at the roof of the neurotube and adjacent to the hippocampus. And then we have the antihem, a patterning center that sits at the boundary. This is the boundary of the pallium and the subpallium, cortex and striatum. It sits right here, and it secretes a morphogen known as PAX6 that opposes SHH. And if you altered the morphogen activity of this area, you can move this boundary back and forth. In the cortex, it is opposed by morphogens from the hem, particularly EMX. And so they diffuse, EMX diffuses from the medial caudal to the lateral frontal to oppose this. And you see the amygdala here. The paleoamygdala is part of the cortex-like amygdala components is part of this model. The other thing is that now going back to Sanidi's model of brain development and evolution, this basically confirms his hypothesis of how the cortex was derived. Basically, again, this is the hippocampus, olfactory cortex, these cortices being allocortex, poorly laminated, and that the gradient of lamination proceeds from these two main areas to establish these cortical rings. And you can see it with the isocortex and the primary sensory areas being little islands within surrounded by these rings and then surrounded by the very primitive cortices. And so this is basically showing the direction of laminar development. So going from allocortex to eulaminate or the well-laminated cortices, typified or exemplified in the primary sensory areas. So the question then becomes, if we have this gradient being just three layers, three-layered cortices, how do we end up with a six-layered cortex? Based on gene expression and the gradients of gene expression and the primitive organization of the pallium, one hypothesis is that these signaling factors during evolution sort of spread into the dorsal pallium, medial pallium, hippocampus, ventral pallium, paleocortex. These morphogens diffuse into the dorsal pallium and establish a six-layered cortex. So the simple summary of this model is that layers 1, 5, and 4 come from the medial pallium. Layers 2, 3, and 4 come from the ventral pallium, stacked on top of the medial pallium. And there's really compelling evidence now for such an organization, because if we look at in studies, we alter the activity of either PAC6 or EMX, we get changes in the development of cortical layers that align with this model. And so from the prosomeric model, the top of the hierarchy, the implication is that the limbic cortices sit at the very top. From a structural model of cortical connectivity, the structural model is used to predict how different areas are connected to each other. And it is based on laminar complexity. If you know the laminar complexity of a particular region, this model allows you to predict how they should be connected. So if you're going from simple cortex, in this case, dysgranular, very little evidence of layer 4, you see connections arising from the infragranular layers, projecting to the supragranular layers of more well-laminated structures. And then the opposite, from well-laminated to less laminated, goes from supragranular to the granular layer. If they are similar, they will have a columnar connectivity pattern, meaning that they span the different layers. And here is just how we would, or shown from a flattened perspective. And so the neural hierarchy here is, again, the top of the hierarchy would be the primitive layers. Okay. So switching to cybernetic control, feed forward and feed back. So in terms of the language, the way anatomists describe connectivity is that going from lower levels in the hierarchy up the hierarchy, the pattern of connectivity is described as feed forward. Whereas from high to low is feedback. And this is sort of like a bottom up, top down description. From a cybernetics perspective, and this is an unfortunate thing because the language is the opposite. In cybernetics, feedback is action, describes actions that are controlled by feedback from the environment, as an example. Feed forward is sort of like a plan of action, and that is projected or executed independent of what's going on in the environment. And so, in this sense, feed forward is actually from the language of, let's say, active inference, it's predictions, feed forward predictions that go into the supergranular layers, and the supergranular layers provide feedback. So for us, this is the language we're going to use, the language of cybernetics. And so for active inference, predictive coding, these patterns basically describe the anatomical structure of predictive coding as described by Bastos et al. In subsequent work. Okay, so now I want to talk a little bit about the evolution. Based on the evolution of active inference, based upon the evidence of the prosomeric model. As I alluded to earlier, this segment here, so we can again see all of the primitives of the mammalian organization here, but the diencephalon and mesencephalon, the thalamus and midbrain, were fused together. Through evolution, during the course of evolution, we believe this is one of the major events that occurred that allowed for the rise of these dual systems. So first, there was a separation of the midbrain from the diencephalon. The diencephalon being the thalamus and the prethalamus. We'll talk about that in a little bit. But the thalamus, the importance of separating or having a thalamus is captured here in the colo and lemnothalamic organization of the thalamic nuclei, the dorsal thalamus, into a group that is referred to as the colothalamus and the lemnothalamus, or another called the lemnothalamus. And so we have the colothalamus being the recipient of sensory signals that have passed through the colliculi and then relayed to the brain. Whereas the lemnothalamus receives sensory input that avoids the colliculi, the midbrain, and goes to the pallium. And this exists in in vertebrates, in all vertebrates. And the relation between these groups to the pallium is as follows. The lemnothalamus has, again, direct projections to the pallium, particularly the medial and dorsal. And this is in a model of a primitive vertebrate. The colothalamus goes to the striatum. In mammalian evolution, this was extended. The colothalamus extended its projection to the lateral aspects of the pallium, with some remaining projections to the striatum. And so this carries from the midbrain, being colo, all the way to the pallium. The importance of the midbrain then and the collothalamus we can see by to cortical evolution by looking at two particular midbrain nuclei that constitute the midbrain locomotor region, the pedunculopontine nucleus and the cuneiform. And these are the nuclei that controls motor activity in response to sensory stimuli and encapsulated within the midbrain. We can see here these are the inputs, the number of inputs, number of outputs. That if we look at the number of inputs, it uh, is centered on the midbrain. And then if we look at the outputs, the cuneiform, the green here, it remains somewhat restricted relative to the PPN. Or much more limited. But when you look at to these midbrain structures, when you look at the PPN projections, it then extends into the forebrain, basal ganglia, and cortex. It's as if there is an extension now of the midbrain motor control system, as exemplified in the motor the midbrain locomotor region, the MLR, into the basal ganglia particularly. And then the diencephalon also in evolutionary development had what we now come to recognize as the prethalamus, this segment here. And the prethalamus is made up of a very small number of nuclei. The two important ones are the reticular nucleus and the zona incerta. These are nuclei that are predominantly GABAergic in nature. And so it being sitting in the alar plate is sort of an unusual situation because SHH, which guides the development of GABAergic neurons, should stop here. So how did we get GABAergic interneurons up here that make up these nuclei? The answer really is quite simple in that the morphogens that oppose the SHH get suppressed and allowing SHH to come into this area to guide this development. During that development, it seems, based on connectivity data, that the zona incerta, another locomotor region, traditionally referred to as the diencephalic motor region, seems to be connected preferentially with the medial prefrontal cortex, the cingulate cortex here. This is just the number of labeled connections going from the cortex to the zona incerta. And so up to this point, I'm trying to lay out the picture of how these areas are aligned with each other to form a system. So here's sort of a summary, a quick summary. If we look at, again, the brain, the cortex, and according to the lemno-colo model, as well as the prosomeric model, we can draw a line here. This is the ventral lateral. This is the medial dorsal, and we can group all those cortical regions into these two trends. And this reflects the organization of how the thalamus is connected. The two components of the thalamic groups connect to the cortex. Okay, so what is then the evidence for separable systems? I'm going to go through this somewhat quickly because I know I have about 5 to 10 minutes left. So now back to cybernetic control. The classic observations, really, we think this is exemplified by the Kluver-Bucy syndrome, which is a collection of symptoms when you have lesions to the anterior temporal lobe and the amygdala, particularly bilaterally. In humans, is quite rare, but you could see it in both humans and animal models, exemplified by loss of fear, hyperorality, basically mouthing objects, hypersexuality, like mounting members of the group, hypermetamorphosis, which is a abnormal distraction by sensory input, particularly visual inputs. We have interpreted this to reflect the function of a medial dorsal system that underlies feed-forward control without the influence of the anterior temporal amygdala system involved in feedback control, basically constraint on action with a valence, a negative valence of fear, anxiety. And what we see here is just the release of the feed-forward system. Akinetic mutism and akinesia occurs when you have lesions, again, particularly bilateral lesions to the medial frontal cortex. It's characterized by poverty of spontaneous speech and motor activity. So we see this as a complement of each other. Some modern observations. So these are studies that use either lesion or temporary functional manipulation of inhibition or excitation. If we look at the hippocampal medial prefrontal cortex circuit bridged by the nucleus reunions, which doesn't project to the basal ganglia, we see that lesions to components of this system produce Kluver-Bucy-like syndromes, particularly enhanced fear, showing the function of the ventrolateral system. And perseveration errors, continuing to choose the same non-rewarded options or ineffective search strategies. The other is a system involved in information novelty seeking. And this is, they're describing basically the Kluver-Bucy syndrome, but in animals. When presented with novel objects, animals explore them by sniffing, carrying, grabbing, and biting the object. And the duration of this is much longer for novel objects than for familiar objects. And we could stimulate structures of this circuit to increase the likelihood of these behaviors. And again, it's very similar to what is seen in the Kluver-Bucy syndrome. And this is centered on the medial prefrontal cortex and the zona incerta. The zona incerta being particularly important here because it is GABAergic and it inhibits fear-related responding in the lower structures like the midbrain and the midbrain structures such as the periaqueductal gray. And then at the telencephalic level, it is the interaction between the amygdala and the prefrontal cortex. We can see a very similar pattern repeated, meaning the medial prefrontal cortex is involved in sort of exploration like activities. In this case, if presented with decision-making under certainty and the context of risky choices or safe choices, if you inhibit the amygdala projections to the medial prefrontal cortex, animals take more risk. If you lesion the prefrontal connections to the amygdala circuitry, you produce perseveration errors. So let me just close here by, again, summarizing or making a particular remark about active affordance. So for us, active affordance has two forms. That is a exploration form and an exploitation. Exploration being motivated and guided by the feed-forward cybernetic control system. Exploitation by the midbrain and amygdala at the telencephalic level. And because they are motivated systems, what are what is the root, or what are the roots of their of these systems? We go back to the prosomeric model to understand that the telencephalon and both of these systems, as reflected in the telencephalon, arose out of the hypothalamus. And from the prosomeric model, the implication is that the hypothalamus sits at the very top of the hierarchy, actually, the neurohierarchy, to motivate behavior. The telencephalic structures, those that have the densest and the closest connections with the hypothalamus are the amygdala and the medial prefrontal cortex, prelimbic and cingulate cortices, and those, as I've shown, are the limbic basis of what we would consider active affordance. But now we have it being traced back to the hypothalamus at the highest level. And so, in closing, that to us sort of explains the model initially put forth by Gary Goldberg as pictured here. So thank you for your attention, and I guess it will be time. It's time for questions. I hope I was clear, but let's see.
Thank you, Phan, for this tour de force. I think it's about time we give you a round of applause. Unmute ourselves and give Phan a round of applause. Excellent. Now we open the floor for the Q&A. And let's see if there are any questions from the audience. All right. So until people collect their thoughts, maybe I can venture one. This is definitely not within my realm of expertise, but I try to follow as much as I could. So one thing that I'm curious to find out, and this is not in any way I would suppose an intellectually stimulating conversation, but it's more for my curiosity. What's the end game for you in reviving this model and proposing this kind of cybernetic interpretation of it? What do you wish to accomplish with this?
So really understanding, okay, let me, I guess, back up a little bit.
The state of the literature, the state of the art in the literature that you presented and all that. So that's the kind of the context.
Yeah, let's see if I can answer this. So, you know, for us, for Don and me, one of the things that has been really influential over the last couple of years, ever since we discovered active inference, actually, is active inference gave us a formal way of talking about ideas that we've had before about these control systems in a much more concrete and generalizable way. And also, it gave us a way, the active inference, a way to understand brain function at multiple levels. And so then the exercise, at least for us, became, how do we then keep pushing that understanding all the way down to what we call active affordance? That is the root basis of motivated behavior. Now, from a scientific perspective, to me, when I look at the literature, particularly functional literature, whether it's human neuroimaging or animal data. Without the right framework, without the right model, um, the interpretations can be somewhat ambiguous, even though on the face of it, it seems very logical. An example of it is this. If you look in the literature at how people explain the findings of behavioral uh, uh, modifications or behavioral deficits associated with the zona incerta, as an example. You have vague notions or vague interpretations or descriptions of it being involved in regulating feeding, fear, and so on. And then you have the other side of it, where you have the midbrain involved in reward, positive behavior, etc. On the face of it, because lesions show you symptoms, the description basically reflect the symptoms. But if you approach it from the perspective that I tried to make clear, the interpretations can be actually quite different and consistent across many studies. So let's go back to the case of the zona incerta. Well, the zona incerta isn't really involved, from our perspective, in the control of the basis of fear-related behavior, as an example. Lesions to the zona incerta just sort of unmasks or releases the system that actually controls fear-related behavior. So, to show its true form, in our opinion, of being a feed-forward system that inhibits a feedback system. Now, taking it back to active inference, we can take this all the way up to the cortex to explain the connectivity of the cortex in the columnar model of predictive coding. That predictions come from the primitive basis of feed-forward control. That is, predictions motivated by exploration affordances, which then ties into how free energy can be reduced according to these two forms, because they are separable and have different basis. So that's a long answer to your question, but I hope it sort of it gives examples of why we think it's important.
Definitely helps me a lot, particularly to kind of box your project and you and Don's projects in kind of the literature space that I'm knowledgeable of. So thank you so much. That was very helpful. Let's see if there are any other questions from the audience. We still have some people on. Yes. If not, I think, Carl, this is unusual, but it's your turn. Oh, no, actually,
Oh, I'm
Ronald.
sorry, if I may.
Sure. I didn't see any hands, so I was looking
Okay,
for a hand.
sorry about that.
Go ahead, Ronald. Sure.
So, thank you very much for this lovely talk. So, I have to say that I'm a real big fan of your work. I stumbled on your line of research actually when I took the subregions of the amygdala a little bit more seriously, because I'm actually an fMRI person, do neuroimaging in humans, so we tend to, just mush different brain areas just together because we don't have the spatial resolution. But I found out that we really have to tease apart the amygdala to actually do proper computational modeling of this very delicate area. So my question is about two brain regions and how they are connected to the amygdala according to your model. So one would be how do you think hippocampus and amygdala actually work together? And the other thing, how amygdala and basal ganglia or basal ganglia in total actually work together with the limbic areas.
Okay. Let's see here. we've been working on a section to, in a way, sort of explain uh, the function of the basal ganglia. Let me see if I can sort of articulate it very briefly. Um, we also see dualities in the basal ganglia. We sort of covered this in the 2024 paper with the organization of the basal ganglia, striosome, and matrix compartments being differentially connected with the infra and supragranular layers. In our more recent work, the paper we're working on with hopefully Carl involved here. I'll be sending Carl a draft quite soon. We've extended this to the direct and indirect pathways. The striosome, with now good evidence, being biased towards the direct pathway, and the matrix being biased towards the indirect pathway. And then of course the structural model is really important to this because it predicts how the cortex is connected to these to the basal ganglia, the limbic cortices, the poorly laminated ones, being connected to the striosome, and that's really the traditional description, as you know, is the ventral limbic striatum. And then there's a gradient that goes out to the dorsal striatum. Interestingly, of the inputs into the basal ganglia, uh, particularly strong inputs come from the amygdala to the striosome and therefore the direct pathway, the amygdala and the medial prefrontal cortex. Within the direct pathway, then we have to think about if these are two systems, feed forward and feedback, feedback being the amygdala, feed forward being the medial prefrontal cortex, as they converge into the direct pathway to of the basal ganglia, they're essentially then competing. How do you balance that? The cybernetics of it, the feed forward, the prediction, and the feedback through this circuit. Well, you could do it at the level of the cortex. Again, that's the medial prefrontal to amygdala. And now your point about the hippocampus is for us related to or is the root of the medial prefrontal cortex from the evolutionary model. And its function is bridged by this nucleus reunions. And the exploration basis, the feed forward basis of this system, really for us, it goes back to the spatial functions of the hippocampus. They sort of go hand in hand. So if you lesion this circuit, then you see Kluver-Bucy-like syndromes of exploration, lack of exploration. So that's one level. So at the cortex, but at the basal ganglia level, you have to balance this out as well. For us, the one thing that I didn't talk about is then how these systems can be modulated both at the cortex and at the basal ganglia by the catecholamines, particularly dopamine and norepinephrine. In our model, we have norepinephrine aligned with feed forward control and dopamine for feedback control. And we think that this is what's modulating the balance of these two systems at these 2 points, cortex and basal ganglia. I don't know if that answers your question directly, but that's generally how we think about it.
Oh, yo, that's really fascinating. Cool. I mean, one follow-up question would be, um, you talked about the amygdala. What part are you exactly talking about in your concept? We are talking about the central amygdala here, right?
Yeah, the central so we're talking about the basolateral
Ah, okay.
bec from a particular perspective. The basolateral is sort of considered, as you know, sort of the sensory recipient component of the amygdala. And there's recent work that take an active inference approach to the amygdala. Are you familiar
Oh
with
yeah,
that
the
work?
this might be my paper then, the
Yeah,
this
oh,
paper. Yes.
which I think it was to me was one of the core insights, right? You know that the central amygdala sends predictions to the basolateral amygdala, and so I tried to extend your logic there, going from if that's the case, uh, what is the basolateral amygdala sending to the cortex? Is it predictions, to follow the hierarchy, um,
Mhm.
or something else, or sensory data to, and another example is the medial prefrontal cortex. Uh, and what is the medial prefrontal cortex then sending to the basolateral amygdala? Uh,
Mhm.
so my thinking is this, and because from the work of Helen Barbas, again, the from the structural model perspective, you can sort of predict or connections between the two structures can be predicted by the structural model. So that gave us some clues as to what's happening. Um, Projections from the medial prefrontal cortex to the basolateral amygdala seems to be biased for outputs from the medial prefrontal cortex to the basolateral amygdala. It's denser in that direction, even as it receives projections from the BLA, basolateral amygdala,
Mhm.
um, which then implies that what the direction of information flow is bias from the medial prefrontal cortex, again, layers 5, 6, to particularly layer 5, to BLA. Then we take the logic of the active inference, it's receiving predictions, and we would say feed forward predictions, of the form of the feed forward form, particularly exploration, that have to be sort of balanced by what you would say is the fear related homeostatic predictions from the central amygdala to the basolateral amygdala. So that has to be balanced there. So maybe the BLA is sort of this paleo structure that is mediating predictions, two forms of predictions that has to be balanced. And then if we look at dopamine, and dopamine is particularly important here because then it is the precision tuning of the midbrain, basal ganglia, and the entirety really of the medial prefrontal cortex, because that's one of the odd things when we look at the dopamine distribution in this area. It's in mammals, it's as if this area was sparse in dopamine until you got to mammals.
Mhm.
And then dopamine sort of appeared there. And we think it's particularly important role in playing this balance. So long-winded explanation.
Yeah, but you can't believe how helpful this was. Thank you very much. I will mull a little bit about it, and I might write you an email if you don't mind.
Yes, yes, and I'd like to share our draft with you,
Oh, that
hopefully
would be
soon.
lovely. Mhm.
Yeah, no, that was a great paper. I was at a loss about the how to think about the amygdala. So
Yeah, me too. So after 10 years of waiting that somebody could write finally this paper, I had to do that. Thank you so much.
thank you.
Thank you, Ronald, for a fantastic question. Now, are there any other questions? You can just unmute yourself or unvideo yourself, uncave yourself, as I like to say, and just go ahead. And if not, I suspect now, after a delay, is Carl's turn.
Sure. Is it my turn now?
Yes, with a bit of a serpentine there. Yes.
I see. Very good. Yeah, thank you so much. That was a wonderful and encyclopedic presentation. Um, I think we probably had the best parts of this session in the presentation and the exchanges which we've uh, just enjoyed. But I'll do my best to sort of pick up on some themes from my perspective that may or may not be helpful. But again, just to acknowledge, you know. The, People like me have grown up with brain imaging and think very much in terms of functional anatomy as read from brain activation results. But I do remember my mentors like Semir Zeki saying it's all in the anatomy. I think you've all the answers are in the anatomy, and I think you take that one step further. It's in the comparative and developmental anatomy. Um, However, that does mean that my feedback is going to be slightly naive because I don't have that encyclopedic knowledge that you have. I should also add, if you want to bring these insights based upon this exquisitely detailed and, I repeat, encyclopedic knowledge of the developmental neuroanatomy and also sort of the neurochemistry and presumably also the epigenetics um, to people in theoretical biology. Um, I would counsel that you try to dumb down a little bit, try to keep it, try to simplify things, because people will not remember.
Yeah,
I'll give you two instances of that. The first instance is that I've often, if you remember Leslie Angelida and her dorsal ventral streams when she was working with Mort Mishkin, um,
yes,
that really caught on, partly due to the force of Leslie's personality, but also I think simply because it was easy to remember.
yeah,
That simple idea, that simple blue or red, up or down, dorsal ventral, what where, was this easy for people to understand. Um,
yes,
I, I think you're going to have to play the same game. And I did get a sense there was a sort of unifying simple dialectic here because you started with it and you ended with it. And so my counsel is for the kind of audience you may get, possibly not in cerebral cortex, but certainly in sort of neural computation or um, for people who do not know about neuroanatomy, it may be worthwhile just picking out that really simple dialectic or dichotomy and giving it a very simple name. And so, for example, the what versus where is very memorable. So I think you need the same kind of um,
yeah,
explorative versus exploitative, or internally generated versus externally generated. Something that people, oh, I see. It's as simple as that, is
right,
it? And
right,
then you substantiate that dichotomy, um, that sort of very coarse-grained kind of functional segregation, with all the detailed anatomy, and then you can talk to people like Ronald. But most people are not going to be like Ronald. They're going to be.
right, right.
So that would be one thing. The other perspective is imagine you're trying to lecture to us on how a car works. and and specifically how a car engine works. And you're a mechanic, and you know exactly all the components of a car, and you keep telling people
Yeah.
about the carburetor and the cylinders and so it can be a little bit overwhelming if you actually
Yes.
the detailed stuff. Um, I say that because, it's likely that these ideas will have to be sold to a number of different audiences, and they will call for a different kind of framing, and a different
Yes.
kind of um, a different sort of language. Um, in respect of that, I'm going to sort of pick up on well, I'm now going to come back to you from the other side of the fence, which is basically ignorance when it comes to anatomy, but an investment in the functional side of things in terms of active inference. And it's another side of the fence in a sort of mechanistic sense. So active inference, when you try to look at the sort of substrates that support active inference and active affordance, um, then they emerge from the way that things must be from an information processing perspective. So active inference itself is an application of the free energy principle. And the free energy principle is itself just a physical account of self-organization, very much in the spirit of cybernetics, but slightly more nuanced and actually a little bit simpler than cybernetics. What I'm saying here is that because you don't have any choices. There's no reverse engineering here. You are forced to say at some level it has to behave like this, and therefore the game now is to try and find the substrate that does this because this has to be done if
Right.
this is behaving. So what I'm going to do is just try to pick up on the sort of key architectural point um, that I think will resonate with people in the future. And by people, I mean the sort of the theoretical biologists who don't have a detailed understanding of the anatomy, um, and just point out just one or two papers that um, address similar issues, but from the opposite direction of it has to be like this. Therefore, can we find evidence that it's working like this? And then my hope is that you can use your encyclopedic knowledge to say, yes, this is a little bit naive and it's too coarse-grained, but because I know the anatomy, what they're actually talking about is the amygdala or the central base, or so to actually sort of look at it through the lens of somebody who has a deep understanding of the actual anatomical architecture and say, well, yes, this is a potential candidate, and it's very consistent with this story that we're telling, this basic division between, or well, I'll try and paraphrase how I understood the story. There is this. Well, I'll make it as simple as possible. Perhaps somewhat paradoxically, the older parts of the brain, the limbic bits of the brain, um, are on deep or on top of a hierarchy. Um, and this is important because um, it is these that ultimately govern motivated behavior, either constraining it in some way or releasing it to enable exploratory behavior, or constraining it in the absence of Kluver-Bucy syndrome, um, you know, constraining it at some level, and possibly even a homeostatic level, to keep the system within its essential variables within viable bounds. So for those people who thought that sort of the paleocortex and the archicortex were just really old stuff, and we've now developed lots of superordinate intelligent cortex. No, that's absolutely wrong. It's the other way around. And I think that sort of inversion of common sense as read by people who don't know anatomy, I think that's one of those simple, powerful messages that people can remember. Oh, that's interesting. I didn't realize it was quite like that. Um, and then you can unpack the story at finer detail. And it does resonate. It does remind me very much of this. I don't know if you know Mark Soames, but he's
Yes.
yeah. So he tells a similar story and gets a lot of attention and I think traction by just telling a very simple story. It's all about the subcortex. This is where feeling happens. This is the thing that's in control. You can be completely, you can still have feelings and operate quite happily without a prefrontal cortex. But if you lose these deeper structures, these more ancient structures, then you are compromising things. So, and to a certain extent, one of the papers was just to mention, Mark, in the font hope that you might be able to put a paragraph in just saying that this is, if you like, um, um, a technically well, in fact, Mark's quite technically skilled. He's a neuropsychologist. He looks at brain lesions and the like. But from the point of view of his writings in terms of interpreting, in terms of feelings and motivated behavior and conscious experience and the like, you would be the sort of the anatomically grounded person, but you are telling, I think, a somewhat related story, or it would be really nice if you could explain how you would accommodate his perspective and his conclusions within this strange inversion. I know it's not a strange inversion from your perspective, but for other people's perspective, it could be a strange inversion. Um, and certainly from the perspective, the formal perspective, and I'm now sort of just going to revert to talking as a biomathematician. I think this strange inversion makes a lot of sense, and I just want to explain why. Um, and if you had time, then to go through to the level that you have time and feel comfortable doing the papers with the agenda of saying, yeah, I think I understand what they're saying. And this actually makes sense from the point of view of the conception of the functional anatomy and the architectures that are grounded in the neuroanatomy and the development. Um, Hopefully that will make more sense if I just show the couple of papers I had in mind. Um, if I can grab the screen, which may entail
Okay.
you,
Let me give it up. I don't know if I. Okay, good.
that's good. Um, so I'll just show you. Ooh, where are we? What have I just done?
Let's see. Let me just. Okay, I stopped sharing.
Good.
So
I'm desperately trying to share at my end now. Oh, here we are. Very good. And share. Perfect. Right. I'm hoping that you can see
yes, I
yeah,
see it.
right, right. The first thing is going to be very, very techy, but I think it's quite fundamental. And well, let me just show you. I am aware that this is being recorded, so I'm going to speak fast, and don't worry about taking notes or remembering anything. You can always go back to the recording if you want to. So this is a purely mathematical treatment of something which I think is central to the argument that you've brought to the table, which is an understanding of the functional anatomy of motivated behavior. Now, I do appreciate that you can sort of unpack motivated behavior in terms of homeostasis and allostasis and all that good stuff. But I think there's a simpler, if you like, link to active affordance and active inference that goes along the following lines: that if we are talking about the kind of behavior that is not reflexive and is not sort of homeostatic in nature, but requires an authentic agency and intentionality, then we have to acknowledge that we are now planning towards intended behavior and to have intentions. So now what we're talking about effectively is the substrates that could possibly support the planning, where you evaluate the goodness of your plans in relation to what motivates you. So what we're talking about is the maths of motivated behavior, and it has to be all about planning. So immediately we're in that sort of the medium ventral little arrow part. We're not sort of catching balls. We're now thinking about sort of how we're going to respond in this situation. Well, actually, catching balls is probably a bad example. We're not talking about reflexes. We're talking about deliberative planning, which may be subpersonal under constraints. Um, And from a maths point of view, when you're talking about that kind of policy evaluation and or more simply planning, um, then what you're talking about is scoring the consequences of some action or sequences of actions. And under active inference, um, those can be divided in a number of ways. And I'm going through this in detail because it speaks to something which you recurrently referred to, which was the sort of the distinction between exploratory and exploitative behavior. And from a maths perspective, um, that sort of decomposition into the two kinds of motivation um, is absolutely fundamental to the planning as inference perspective. So this is just the maths behind it. And I'll explain, and try and motivate why I think this is important for the current arguments. Just very briefly, this is the expression for all the sensory hierarchies, all the stuff that might be going on in sort of visual hierarchies, where you're just using available sensory afferents to infer the latent causes to work out the current state of the world. That's very interesting, but not from your point of view. It's got nothing to do with motivated behavior. It's got nothing to do with motivation. It's just um, doing belief updating in a way that you have the best kind of posterior beliefs about the state of the world so that you can then plan. So that you can then um, evaluate different kinds of behavior and select those that are motivating you the most. And that depends upon the free energy that you'd expect if I pursued this particular plan. And there's a nice sort of mapping between the decompositions of the free energy that is being minimized by the perceptual part. And it can be expressed in terms of a mixture of accuracy and complexity, which just means that you can understand neuronal dynamics as representing or encoding probability distributions, posterior beliefs that provide the most accurate explanation for the sensory afferents that is as simple as possible by minimizing the complexity. Um, the nice thing is that when you move to the expected free energy, so now we're talking about rolling out into the future, before I've actually acted in order to evaluate my plan. Does it do what I intended? Or can I score the goodness of this particular plan? Then the expected complexity becomes risk and the expected inaccuracy becomes ambiguity, which basically means that the most likely policies pi are those that minimize risk at the same time as minimizing ambiguity. And risk is just basically the difference between what I think is going to happen, the states of the world I think will happen if I pursue this policy relative to my preferences, my prior constraints, those things that define the states that define the kind of thing that I am. And I'm going to minimize the divergence. And this is what this is all about here, basically, just unpacking this, but we don't need to go through this in any detail. Um, um, but at the same time, I'm also going to minimize the ambiguity of my observations, my primary sensory afferents, in the sense that I am going to be moving towards the light. I'm not going to cover my eyes. I'm going to be looking where I know that there's some salient, unambiguous information, and these two things, interestingly, can be well. We can swap around the constituent terms of the risk and ambiguity and get something called extrinsic and intrinsic value. And this herein lies this fundamental divide between the exploration and the exploitation. And the exploration is just simply the degree to which I change my mind if I commit to this act, or the degree to which I think I will change my mind if I do this policy, afforded by the outcomes and the observations that I would get if I did this. This is mathematically just scores the KL divergence or the information gain with and without the outcomes I would observe if I committed to this policy and the information is about the states of the world. So you can read this in many ways. It can be read as a salience map. It can be read of pi. It could be read as information gain. It can be read as epistemic affordance, intrinsic value, intrinsic motivation. In robotics, they call it intrinsic motivation, which is why you'll be perfectly entitled to say that the motivations or the likelihood of a particular policy is simply a mixture of its intrinsic motivation and its extrinsic motivation, where this extrinsic motivation is just the the probability that the outcomes will be unsurprising for me. So it's a negative log probability, so it's a surprise or a self-information term. So this is what basically affords the constraints on my exploratory behavior. So these constraints are the ones that keep in check my exploratory behavior, exactly in the way that you were describing in terms of there are two kinds of the amygdala keeping things in check, in the sense that I am not at liberty to explore everywhere unless I have Kluver-Bucy syndrome, because I have these homeostatic constraints, these innate preferences, and other preferences that I can learn as I grow up. And it strikes me that this mathematical architecture fits very beautifully with your notion that there is going to be a very delicate balance between the intrinsic and the extrinsic motivations that depends upon the precision of your preferences. And the key thing that comes from this mathematical analysis is that the balance between the intrinsic and the extrinsic motivation emerges from these top-down constraints that afford your prior preferences. So exactly as you're suggesting, the parts of the brain, they're going to provide the predictions in the form of prior preferences or constraints on your evaluation as enumerated by the expected free energy must be at the top of the hierarchy. So in this view, you can think about a hierarchy from a maths perspective as providing these top-down predictions that are in a predictive coding scheme complemented by the feedback or the ascending or bottom-up prediction errors. That's a good model for continuous state space models and has a lot of currency in relation to predictive coding, Kalman filtering, Kalman-Bucy filtering, and the like. This calculus, though, is more general. This is called belief propagation or variation because it's about discrete states of being. I am hungry, I'm not hungry, I'm in the kitchen, or there's a tiger there. So this is all inference, which is not so much framed in terms of predictive coding, but has the same the same calculus behind it, where we still talk about precisions, but now they are precisions which are negative entropies or confidence that you describe to this or that. And on that view, you can look now at the top-down predictions or the descending predictions as providing mathematically what's called empirical priors. Some people in machine learning call them inductive biases, but you can also think of them as constraints. So basically, you can look now at, say, the amygdala as providing top-down constraints on the evaluations of policies in the basal ganglia that are then used to supply predictions of what I'm actually going to do to the cortex and to all the way down to the neuromuscular junction in a sort of proper cybernetic control. So it struck me that the story, the strange inversion story, and putting these ancient bits of the brain on top because they have to be on top because they are providing the constraints that underwrite the extrinsic motivations that determine the degree to which you can be the degree to which your intrinsic motivation, your exploratory tendencies can be expressed or not, that underwrite your policy selection in the, say, the basal ganglia. And now you can also equip, if you want to, as you indeed mentioned, you can sort of contextualize your prior preferences, these innate constraints on the kind of outcomes, usually of a sort of interoceptive or homeostatic sort, that characterize your physiology and your phenotype, equip them with neuromodulation in terms of that can make them more precise. So there will be some situations where, this particular kind of outcome is going to be very precisely controlled. In other situations where I won't mind, and I can sort of modulate that through the release of dopamine or noradrenaline. So I'm putting that forward to you, just to, in a game of tennis, you're throwing anatomical tennis balls to me. I'm throwing maths ones back to you and see if you can make sense of them. And if we bat to and fro, then we can have a beautiful synthesis.
Yeah.
Yeah. So that was this paper, which
Okay.
there are lots of descriptions of this, but this is given your relationship, your role, and working with Don and people, I'm sure you have access to. You will get into the maths of this because it's relatively straightforward. So that's good. If you take that maths and just ask now how it would unpack in purely mathematically. So there's no anatomy here, but there is a sort of a cybernetic starting point, which is that you're going to assume it has to be the case that your forward model, your world model, your generative model has to be a good regulator in the spirit of Ross Ashby, in the sense that the structure of your generative model has to in some way mirror the structure of the controlled environment, be it your motor plant or be it your sort of extrapersonal environment. And if you accept that, then it means that the anatomy, the mathematical anatomy at least, has to have some deep, or has to have a hierarchical depth, and therefore has to have a deep structure. Simply because the world has a separation of spatiotemporal scales and has this deep structure out there. So I know that my generative model has to have a deep structure. And if I just run through either discrete state-space models or continuous state-space models, depending upon where I am in this hierarchy, and think about the implications of having a state-space model here in terms of a Markov decision process, and here in terms of continuous variables, and then I now build the next layer, which is usually universally mathematically slower. So the layer above usually sort of ticks over and updates at a slower rate than the level that is in exchange with the sensory motor input. So again, that's consistent with this notion of the deeper levels of a hierarchical structure, providing inductive biases or empirical priors that place contextual guidance or constraints on what's going on before, noting that the context has to outlive in time the content. So it's always the case that the deeper top-down parts of or predictions or constraints or inductive biases are always going to be, if you like, changing more slowly than to contextualize a faster activity, sort of catching the ball or some sort of fast autonomic reflex. And that's just sort of unpacked mathematically here. But when you put the motivated behavior into the game and you start to think, as this thing does, then you, this is the equivalent Bayesian filter, or the equivalent for continuous state-space models, and some laminar, ulaminar, or possibly dysgranular representations of that. When you put motivation into the game, then you start to think about, well, where and how, given the constraints on the belief propagation implied by these deep generative models, how would you have to gather all the requisite messages to actually evaluate the expected free energy, which balances the exploratory and exploitative aspects of motivated behavior? And interestingly, um, so this is where somebody with anatomical naivety comes into play, because they just look at the maths and say, okay, given my sort of student level knowledge of anatomy, what fits in terms of the connectivity. And this is, one attempt at this jigsaw, which you are in a much more powerful position to actually put together properly. But it does strike me that there's a possibility that this could be unpacked by an expert, i.e. you. Um, 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 Fairly simply, because even with my knowledge of anatomy, I was able to realize that all the bits of the brain that you were talking about are really important when it comes to the actual evaluating the motivated behavior, and indeed sort of focusing on the message passing, in terms of cortico-basal thalamic loops, and having that sort of spiral architecture. I can't remember what it's called now, but sort of, deep as you get more towards the medial prefrontal and the frontopolar regions, a different sort of scale, time scale emerges. So I was hoping that you could look at this figure here and think, yeah, he's got the wrong anatomy, but the spirit was right, and it fits exactly with the story that we're telling, which is that these motivated behaviors that rest upon a generative model of the future, because it has to intend to do something means I have to have a representation of the future outcomes that are my intentions. We need this special anatomy that needs to be constrained, and it needs to be constrained by all the subcortical structures, and also involve the precisions. Although I don't think the precisions are actually in this particular um, diagram. So that was my fond hope there. Um, d- d- just to sort of continue that theme, I repeat, you've already mentioned sort of the precision of your beliefs about your plans and your intentions being a really important modulator of action selection. Um, and I noticed that you were talking about notions of the both the differential fields of termination of the different catecholamine or ascending classical modulatory neurotransmitters, and also, interestingly, the sort of developmental changes as we go on. In the limited but possibly um, uh, in quite important space of discrete state space models, most of the um, empirical evidence suggests that the dopaminergic system does exactly as you were saying, sort of encoding the precision of beliefs about policies. So we're talking now about representing literally, I can pursue this plan or this plan or this plan, and the relative activations of these things is going to be determined by the precision. And if I'm very, very confident this is what I'm going to do, I'm going to commit to this plan. I feel good about that. Loads of dopamine, lots of very precise, winner take all like stuff, and with lateral inhibition, all the things that are not going to be selected. Then in this again, coarse-grained, almost cartooned deployment of neuromodulators, dopamine clearly encodes the precision of beliefs about policies. Noradrenaline or epinephrine
Mm.
looks to be much more interested in the dynamics. So again, when talking about sort of motivated behavior that has this intentional aspect, you're talking about a generative model that can generate the consequences of one action or another action. And that
Mm.
means that you have to have a generative model of dynamics and how things unfold in time. But of course, you're going to accrue uncertainty as you move forward in time, and you can think of it like reverse working memory or pre-play activity, for example, in planning. And it looks as if the adrenergic system seems to encode the specific uncertainty or precision of these dynamics. So if I'm very confident what's going to
Mm, mm,
then I would have a high precision, very unconfident about the, how things are going to unfold. If in a very high volatile environment or in a predictable
mm,
environment, then
mm,
I need to adjust that. And then finally, it looks as though cholinergic that sort of controls the precision of likelihood mappings, which is basically the degree to which I trust my sending messages. That do
mm,
my brief updating. So it's much more on the sort of, well, not just on the sensory side, but the mapping between one level and the next. How much do I trust the level below? Um, uh,
mm,
as opposed to within this level, how much do I trust my estimates of the future? As opposed to with dopamine, how much do I now trust my estimates of what I'm going to do in the future? So that, there's a nice sort of decomposition in this mathematically constrained um, approach. So I thought that I mean, it's the next level to the story. I think you can tell
yeah.
the anatomical story without even mentioning precision, but it's such an attractive thing because, of course, the origin of most of these um, incredibly important control parameters is intimately related to the systems that you've been talking about, and of course, intimately. Sorry, so that is that paper. No, sorry, that table and a brief discussion of the empirical evidence that sort of ties these different neuromodulatory neurotransmitter systems to different kinds of precisions was written by Thomas Parr, Royal Society Interface, Uncertainty Epistemics and Active Inference. Um, and then this is just an arbitrary paper by Mark Soames, just to say, of course, all of his story is about the encoding of that precision, which he associates with felt uncertainty. And it's all in the subcortex, it's all in the brainstem. For him, the cell, he goes on about the reticular activating system, but I think he means sort of VTA, nucleus basalis, and the cells of origin of the nucleus raphe, the cells of origin of the neuromodulators. Which now begs the important question: what controls them? And I would imagine you know exactly what controls those, and it's exactly these things that are on the top of the hierarchy, um, and they have to be because this precision provides a context. So it has to be a top-down contextualizing effect, deep from deep within the hierarchy. Um, so I just want to mention that, and then finally, yeah. So the author of that paper, just thinking about the selective roles of neuromodulators, also worked with um, people that you may have come across, particularly Mike
Yes.
Hamilton. Yeah. So I think I don't know, he didn't actually go to visit, or perhaps he did for a few weeks. I'm not sure. Anyway, he was just trying to um, model their data um, from optogenetics of mice doing quite a sophisticated sort of planning and working memory task, looking at sort of prefrontal thalamic interactions, and asking could he model that in terms of exactly the mechanics that we've just spoken about. I don't know whether this is going to be useful for you, but it did strike me as a sort of worked example of applying that maths to to an empirical setup which touches on some of the particular connections that you were interested in. So this may not be interesting, but it is an example of how one could apply the maths to the components of the system that you were focusing on, or you could coerce it to be so. Um, and finally, I just put this up just because I think this is the first time that I was a co-author on a paper claiming that the hippocampus was at the top of the hierarchy, uh, which again for me, it took me years to realize that. For many people, they still haven't realized it. But I was reminded of this work. Simply because of your focus earlier on in terms of the epigenetic or morphogenetic control of GABAergic neurons from the basal plate. So Helen is she's in Oxford and is an expert in functional anatomy of the hippocampus, but had a particular interest in the role of inhibitory GABAergic neurons and their role in providing these top-down contextual guiding constraints. Again, it doesn't fit with the specific proposal that you need to communicate, but it is something that is entirely consistent with as an example that you can point to say, look, even when you unpack it in this instance in the context of predictive coding. So her examples here, yeah. So she's predicating this not on this belief propagation scheme, which Thomas Parr was using, but more in terms of a predictive coding scheme where the precision now simply boosts prediction error populations in the supergranular layers. But her focus really was on the importance of inhibitory interneurons in mediating the descending contextualization and control of cortical representations or dynamics at lower levels. That may or may not be interesting, and that's it. That's
Well, thank you. As usual, I'm struggling to keep up with your explanations, but I see the core parts of what you're saying. So I'm gonna struggle to hang on to those and work through the papers on that. And in some ways, you're the Parr and Friston paper on the basal ganglia. Really, I struggled to understand that. I don't know the math, but it really guided my thinking about some of these the points you've made, and I didn't discuss that today. But the notion of action selection through the basal ganglia, as outlined by Parr and you, really guided my thinking on a lot of these. So please forward these papers to me. I'll struggle through them. I guarantee I'll struggle.
well, now you know how I felt when you were doing all the anatomy.
Yeah, and your recommendation also about, getting a main message is right as well. Don and I have gone through multiple cycles and conversations about this, and we're arriving at more or less your recommendation, but then the struggle is how to simplify it. And of course, we're relatively naive to all your points, but I understand the importance of it. I grasp the importance of your points, and so it's going to be somewhat of a struggle for me, and hopefully Don can help me as well integrate I love your points about the intrinsic and extrinsic components here. So I'm going to try to hang on to those, what I can remember, but hopefully I can have a couple of Don and I can loop through several cycles with you on some of these with the paper we're preparing, and really get your guidance as you're saying here on some of these. So nuggets, what I can, valuable nuggets, what I can hang on to. Thank you very much.
No, thank you.
Well, we thank you, Phan, for this fantastic tour de force and encyclopedic presentation. And if there are no other questions, we give Phan a final round of applause.
Thank you for the opportunity.
Yeah. All right. So that concludes our meeting for today. Carl, could you please hang on for 5 minutes to clarify that administrative wrinkle with you being able to open the presentation? Thank you, everyone. Feel free to log out.
Thank you, everyone.
Bye. Thank you, Phan. All right. So what I will do now, you have the link for the meeting, right, Carl? You are muted. Carl, you are muted.
Yeah, what I'll do is I'll just leave. Why don't we both leave, and then I'll try and start it,
And
and
see if
then you join me. And if that doesn't work, wait 2 minutes, and then I'll join you. So
all right.
let's.
All right.
Yeah, brilliant.
Great.
Right, see you in a
Bye.
second.